Digestion is a term used to summarize the complex coordination and regulation of a number of processes involved in moving nutrients, water, and electrolytes from the external environment to the internal. These processes involved in digestion include mechanical and chemical breakdown of foods, absorption of the products of this breakdown by the GI epithelium for distribution throughout the body, secretion, which can have a couple of meanings. One, referring to the movement of water and ions from the interstitial fluid into the luminal space of the GI tract, or another meaning the release of substances synthesized by the GI epithelium into the luminal space or into the interstitial fluid, respectively. And the final process involved in digestion is motility, which is the movement of materials throughout the GI tract via muscle contraction. These final two processes, secretion and motility, are the most highly regulated. And it is through this regulation that maximum absorption of materials as well as the reduced waste of secretions can be maintained. And it is through these processes collectively that a number of challenges involved in digestion are faced and dealt with. Some of these include digestion, maintaining mass balance, as well as defense from microbes that enter the GI tract. In terms of regulating the digestive process, there are a number of different signaling molecules and pathways. The first is a series of peptides called GI peptides, which may be secreted into the stomach or into the interstitial fluid by the GI epithelium. There are also a number of hormones that fall into one of three categories that are involved in signaling cells involved in digestion, as well as a system of nerves known as the enteric nervous system, which involve a few ganglia and a number of sensory and efferent nerves that will regulate the function of the tissues involved in GI tract digestion. The enteric nervous system is said to be the small brain of the human body because it involves both long and short reflexes. Long reflexes meaning that sensory information is received, it is then passed to the central nervous system, and the efferent nerves transmit signals to peripheral ganglia and eventually reaching their targets to have their effect. However, within the enteric nervous system, there are certain signals that do not require the input of sensory information to the central nervous system. Instead, they have what are known as short reflexes, where sensory receptor cells receive sensation. These sensations then synapse in enteric nervous system ganglia, and their effect is then felt through the efferent nerves of this enteric nervous system. Digestion can be divided into one of three phases: cephalic, gastric, and intestinal. Specifically discussed here will be the gastric phase of digestion because it is one of the most highly regulated. However, before we can talk about the gastric phase of digestion, we need to mention the cephalic phase, which is a period of digestion that occurs before food is even eaten. This is an anticipatory sense or thought of a meal that may stimulate the digestive system. Either smelling, thinking, or seeing food could simply trigger the cephalic digestive system, the cephalic digestive stage. And it is this cephalic digestive stage that will work through long reflexes to stimulate the enteric nervous system to prepare for the incoming meal. The digestive phase, or the gastric phase, involves the stomach, and specifically the stomach has three important roles. It is involved in storing and regulating motility. It has obvious digestive function, and it also functions in defense. The anatomy of the stomach is depicted here on the right-hand side. We can see in this cutaway of the stomach that the internal space, or the lumen, is found inside. What lines the walls of the stomach are these wrinkles and folds known as rugae. And if zoomed in close enough, we can see that these rugae actually have openings, these gastric openings, which lead down into these pits, which are known as gastric glands. In these glands, we can see that there are a number of different cell types that make up the gland, and each one of these cell types has a very specific function. This image further enhances that idea that each one of these cell types that make up the gastric gland has a very different form as well as a very different function. Specifically, if we were to look at the cell types that make up the gastric gland, there are 6 listed here in the first column. Their secretions are listed in the second column, and the functions of these secretions are in the third column. Mucus cells make 2 protective compounds, 2 protective secretions, mucus and bicarbonate, which help to protect the epithelial cells from the luminal contents of the stomach. And these cells respond to mucosal irritation as well as secreting mucus tonically. The other cell types listed below all have a very specific secretion. As we can see, the parietal cells make acid as well as something called intrinsic factor. Enterochromaffin-like cells, or ELC cells, produce histamine. Chief cells produce the enzymes involved in gastric digestion, pepsinogen and gastric lipase. The D cells of the gastric glands produce somatostatin, a hormone, and the G cells produce a different hormone known as gastrin. The function of each one of these secretions is listed in the third column. We can see that the parietal cells, the acid that they produce activate pepsin. The intrinsic factor produced by the parietal cells is involved in the absorption of vitamin B12. Histamine is involved in stimulating acid secretion. The 2 secretions of the chief cells are involved in digestion, pepsinogen for proteins, gastric lipase for fats. Somatostatin has a signal to stop the production of acid secretion, and G cells stimulate acid secretion. In terms of how or what these various cell types respond to, that is found in the final column over here on the right-hand side of the slide. We can see that a number of cells respond to acetylcholine, suggesting that they are innervated by the enteric nervous system and respond to signals of acetylcholine, that neurotransmitter. Some cells respond to some of the hormones secreted by other members on this list. So let's take a look at this process in a little bit more detail. The initial stimuli for the gastric phase of digestion, as I mentioned before, can be through the cephalic phase, signaling through one of those long reflex arcs, anticipating the delivery of a meal. This initial stimuli may also be in the form of food entering the stomach. The presence of amino acids and peptides in the stomach, as well as the stretching of the stomach, the distension of the stomach, also signals the enteric nervous system to stimulate some of the cells. The first cell of mention, the cells that respond initially to either of these initial inputs, are the G cells. These G cells in the gastric glands are the producers of a hormone gastrin, which will enter the bloodstream, and it will have an effect on other members of cells within the gastric gland. Specifically, gastrin targets 2 other cell types, those ELC cells, to produce histamine, And additionally, gastrin will also work on parietal cells, and the parietal cells are responsible for acid production. I'd like to point out that these G cells that produce the gastrin respond to the initial stimuli from either food or distension in the stomach or input via the vagal nerve. The product of the G cells, gastrin, then signals the production of acid by directly stimulating parietal cells and indirectly stimulating ELC cells to produce histamine to stimulate the parietal cells. But also notice that the ELC and the parietal cells also both receive and respond to that long reflex from vagal nerve stimulation to produce their secretions. Inside the parietal cells, the way that acid is produced is responding to those various hormone signals or electrical stimulation via the the ENS, is to put ATP synthase pumps into the apical side of the parietal cells. These enzymes specifically pump protons from the cytoplasm of the parietal cell into the lumen, and at the same time bring in potassium ions from the lumen into the parietal cell cytoplasm. Notice that potassium then is able to diffuse back down its concentration gradient back out into the lumen of the stomach. The protons that are used in this pump come from the splitting of water. The other product of this split, the hydroxide ion, is combined with the metabolic waste of carbon dioxide through the enzyme carbonic anhydrase to produce bicarbonate. Bicarbonate then leaves the basolateral side of these parietal cells to diffuse into the capillaries with the exchange of chloride ions into the parietal cells. The chloride ions that enter into the parietal cells then diffuse down their electrochemical gradient into the lumen of the stomach, thereby producing inside the stomach at the end of this function high concentrations of hydrogen ions and chloride ions to produce hydrochloric acid in the stomach lumen. This secretion of acid into the stomach in turn has a number of effects. As shown here in this diagram, in a continuation of the diagram, we can see that hydrogen ions then stimulate through nervous system signaling the final type of cells involved in activation here, the chief cells, to release pepsinogen and gastric lipase. Pepsinogen is inactive in the acidic environment of the lumen. It is then cleaved and activated into pepsin, where it can begin digesting proteins. Interestingly, the production of acid also acts as a negative feedback pathway, shutting down this system, this response to food or vagal nerve input. Acid production stimulates D cells to produce somatostatin, another hormone that has the opposite effect of gastrin. It shuts off G cells, it shuts off ELC cells, it shuts off parietal cells, and it even shuts off chief cells, and when I say shuts off, I mean preventing the release of their secretions, so that this response to food being in the stomach does not continue nonstop. This is a way of regulating the length that the production of these chemicals exists. Finally, this is just a summary of everything that has been mentioned so far. The signals being listed here: gastrin having an activation of ELC and parietal cells, stomach acid activating the chief cells, having an effect of digestion, as well as preventing or ceasing stimulation